remains a mystery. The AIDS test. Let us recall that the AIDS test is supposed to detect antibodies produced by the immune system in response to infection by the virus. This is routinely done by layering proteins ostensibly from the virus in the wells of a plastic rack and adding blood serum to be tested to each. If antibodies are present, they bind to the proteins, and when this happens, sophisticated staining procedures can make this visible. But because no proteins which are viral and free from contaminants have ever been obtained, one cannot be sure what the antibodies are that bind to the proteins. This is the crux of the problem facing all HIV/AIDS tests: the inability to isolate a viral entity and to characterize its constituent proteins unambiguously means that the evidence for the existence of HIV using antibodies is just arguing in a circle. Antibodies that are detected are due to other causes. Why no HIV test is ever able to work? It is consequently quite illogical to claim that a positive test results from prior contact with the virus. 19. Because various ill-characterized proteins are involved, every test kit manufacturer applies his own arbitrary criteria, and no two kits ever give the same result. It makes no difference that learned committees set standards to decide which tests should be regarded as positive and which not, because this merely skirts round the problem, namely, to what are antibodies actually being detected in the AIDS test. It is of no help that nowadays second and third generation tests exist using synthetic proteins which give greater consistency and comparability, because only by an unscientific stretch of the imagination are they viral proteins. Neither fudging the true identity of the proteins, nor advocating two kinds of tests, reassuringly but mistakenly described as search and confirmatory tests, resolves this difficulty. The illicit test is used to screen for antibodies, which is confirmed by the more specific Western blot. The dilemma cannot be stated more poignantly than by quoting from the leaflet accompanying one such test kit. The test for the existence of antibodies against AIDS-associated virus is not diagnostic for AIDS and AIDS-like diseases. Negative test results do not exclude the possibility of contact or infection with the AIDS-associated virus. Positive test results do not prove that someone has an AIDS or pre-AIDS disease status, nor that he will acquire it. 20. Quite the direct proof of HIV. Some HIV researchers have tried to circumvent the problem by pointing to something called direct evidence for the virus. All that this meant, though, was arbitrarily selecting a protein of a certain size which happened to coincide with that shown in HIV models. The delusion of such evidence was illustrated when the protein later turned out to be of human origin. 21. How the genetic information of HIV was manufactured through. Despite this deplorable state of affairs, the majority of AIDS researchers still cling to the authenticity of HIV, because a genetic sequence for it has been published. Moreover, genetic procedures now exist, which, unlike antibody tests, attempt to identify the presence of HIV more or less immediately, instead of only weeks later when antibodies are formed. The fact that the genetic tests, PCR right parenthesis 22, do not give the same results as the antibody tests is simply ignored. Since no virus has been isolated, it follows that no nucleic acid has been isolated from it either. Complicated procedures are even so described in the literature, at the end of which something is produced which is called the nucleic acid of HIV. 23. A test tube. HIV and its DNA can allegedly be made by the bucketful. 24. But under very surprising conditions which, inter alia, entail the use of extracts from plants and other oxidizing chemicals, which could not possibly exist in viable, immortalized cell lines devised and later patented by the Montagnier and Gallo groups, are co-cultured with extracts from human cells or the cells themselves. At the end of it all, HIV itself is not actually obtained, only reverse transcriptase activity is shown to occur, which is taken to imply that the DNA that is found must have been viral in origin. The real explanation of what happens is as follows. In the mixture of cell cultures and stressed human cells, RNA and reverse transcriptase come to be produced in large amounts, because the cells have been specially selected and treated to do this. The RNA is transcribed into DNA by reverse transcriptase, and long pieces of DNA are produced which are said to be viral DNA. In fact, they are composed of unrelated pieces of expressed cellular RNA, transcribed into DNA and linked together by a process of template switching, a well-characterized property of reverse transcriptase. 25. This misleads ordinary researchers into believing that they have actually produced viral DNA. It is said that this linear DNA is the free, or the non-integrated form of HIV, which furthermore is said to be a unique feature of HIV, because a lot of detectable free linear DNA has not been suggested in any other models of retroviruses. And a selecting process. The resulting pieces of DNA too are necessarily both shorter and longer than the correct length of HIV. Pieces corresponding to the correct length of HIV must be selected for size, because otherwise the purported DNA preparation would be a mixture of various lengths, which would violate a cardinal rule of virology, that all nucleic acid of a particular virus be identical in size. And a detecting process. Having artificially prepared DNA pieces of uniform length, they are still not ready for presentation, because they consist of a mixture of all kinds of RNA fragments transcribed into DNA and thus cannot be shown to represent unique viral DNA. Accordingly, the mixture is subjected to a kind of lock-and-key detection process called hybridization, whereby pieces of DNA are detected which complement more or less a probe of that which it is desired to be shown to have been prepared. And choosing a desired probe. Since no DNA from HIV existed to hybridize with the prepared DNA, Gallo and Montagnier simply used stretches of DNA from what they said was specific to HTLV, a retrovirus Gallo had earlier claimed to have discovered, and which they deemed suitable for this purpose. The DNA detected in this way was replicated and certain stretches of it cloned and declared to be the DNA of HTLV3, later to be called HIV. To summarize, the purpose of the exercise is to grow HIV, but it actually produces a mixture of different lengths of DNA, contrary to theory which says they should all be identical and no virus at all. It is then claimed that the correct DNA has been prepared by finding certain strands in this heterogeneous mix, by hybridizing them with an HTLV DNA probe whose sequence is known and defined to be similar to HIV. However, non-hybridizing strands of DNA should not be there at all, and the fact that they are proves that a complete ragbag of DNA has been prepared, without any indication of what it is made up of. It follows that HIV DNA must just be a laboratory artifact constructed to a preconceived idea of what retroviral DNA should be, and this assessment does not even raise the question why no virus can be obtained, whatever the experimental conditions. Gallo and Montagnier's cloned HIV DNA. One cannot help asking why no one had not long ago spotted the flaw in the techniques employed by the Gallo and Montagnier groups. After defining some segments of DNA to be HIV-specific, every researcher in the field worked exclusively with short clone sequences, never the whole strand, on the reasonable assumption that the original characterization had been correctly performed. From the isolation and identification procedure described above, it follows that the resultant sequences vary widely from one preparation to the next, which sequence analysts misinterpreted as the legendary capacity of HIV to mutate. A computer-simulated phylogenetic tree was constructed, which established precisely what its designer sought to prove. 26. Some history. Perhaps one reason for this calamitous state of affairs is that HTLV3 was presented to the world as the cause of AIDS at a historic press conference on April 23, 1984. A patent for an antibody test was applied for on the same day, instead of making the evidence for it available beforehand, as correct science demands. The undue haste may be explained by the fact that both the National Cancer Institute and the Centers for Disease Control, CDC, had actually one day earlier in a lengthy front-page article in the New York Times on April 22 come out in favor of the French claim for priority. 27. 2. Even so, one must admire Gallo's audacity, because using the same technique